Um, Can you hear me? Good. Yeah, I don't know, it doesn't. Oh. I'll try that. Can you hear me now? Okay, so before I introduce today's speaker, just a reminder, our next speaker on March 27th, that's 2 weeks from today, will be Professor Richard Bark from the School of Public Policy, so a little bit of a departure from our traditional technical talk. We'll be hearing more about kind of policy issues related to nanotechnology. So for today's speaker, it's a real pleasure to introduce Professor Shuichi Takayama. Professor Takayama is relatively new to Georgia Tech. He got his bachelor's and master's degree at the University of Tokyo in agricultural chemistry, and then his PhD at Scripps Research Institute in chemistry and chemical biology, and then did a postdoc at Harvard in the same areas before spending 17 years at the University of Michigan in the Department of Biomedical Engineering. So what do I always say when we have a speaker who has Michigan ties? Does everybody remember? Go Blue. Particularly this Thursday at 9:50 p.m. He came to Georgia Tech last year, where he is, as a Georgia Research Alliance Eminent Scholar and Price Gilbert Chair in Regenerative Engineering and Medicine, and a professor in the Department of Biomedical Engineering. In addition to founding a couple of companies, he's also a winner of an NSF Career Award and several others, and is serves on editorial functions on journals as well. So it's a real pleasure to have Professor Takayama.
Thank you. It's great to be here. Thank you for the invitation and opportunity. And this winter's been great. Coming from Michigan, it's nice and warm for us, although I hear that it's kind of cold for Atlanta. So my talk today, I want to tell you about some microfluidic devices we make with inspiration from electronics, the ability of electronic circuits to do interesting switching operations. We try to adapt to microfluidics, and I want to describe some of the device aspects of what we do in terms of microfluidics. And then, coupled with that, some of the biomedical applications. I also want to point out that I don't have an acknowledgement slide at the end, but I have a list of names and collaborators that helped out during the project, so I want to acknowledge them here. Okay, so first of all, motivation for why we do what we do. So this is a picture of bacteria. If you were alive 100 years ago, your major health concern would be early death from infection by pathogens like this. So how did we overcome this? Many things, but one thing that was helpful was to grow the cellular causes of disease outside of the body, often in dishes like this, and then study what these cells do and come up with ways to prevent what they do, develop therapeutics and so forth. And this was very successful. Now today, our major health concern is chronic disease, like cancer, cardiovascular disease, and so forth. Accounts for a majority of deaths and account for a majority of healthcare costs. So what can we do? How about trying to bioengineer or recreate an aspect of these diseases outside of the body as well? So we might say, well, let's culture the cells that cause chronic disease. And these cells would be not bacteria, but cells in your body, mammalian, human cells. And here's a picture. Now, a little bit about the size scale. These cells are on the order of tens of micrometer. And just for comparison, this is the picture of bacteria that I showed a few slides ago. So in terms of volume, our cells are orders of magnitude larger, and they have complex behaviors. They try to not just grow a lot, but coordinate functions with each other. So even though we can readily these days grow human cells outside of the body in dishes like this. When we do so, their behavior may not reflect what the same cells do when they're inside our bodies or in diseases. And so when we try to use cells grown in a dish to understand basic cell biology or test therapeutics or so forth, bad things or wrong things can happen or be misleading. So my lab and others try to overcome this physiology gap with new ways to culture and study cells. And this has given rise to a field of so-called organs on a chip, tissue on a chip systems, where we grow cells not just in static dishes, but in microfluidic systems and with dynamics that might mimic a little better what goes on in the body. So let me ask you a question. If you want to check, or maybe I should step back. So what is our goal here? So our goal here is we want to kind of breathe life into these organ-on-a-chip systems so that cells behave physiologically. Then I want to ask the question, so for a person, if you want to check if a person is alive, what do? How would you check if a person is alive? Maybe you check for a heartbeat or check if the person is breathing or not. And in microfluidics, this is like having pulsed fluid flow. Mechanical and chemical pulsing are an important part of physiology. And in the body, it's not just the heart and breathing that give rise to pulsing flows. It's also things like the oviduct, our neurochemical signals are pulsing, and so forth. So our motivation for developing microfluidic pulsing, microfluidic switching circuits is to try to recreate an aspect of this outside of the body. So now let me go to some specific applications. So the first one is about pathological pulsatile flow in the lung. So most of us have gone to the doctor and had the doctor listen to our chest with a stethoscope. I don't know if you've ever actually listened to these sounds. So all of you look very healthy, except for I heard a cough somewhere. But most of you, your lungs should sound like this. So this is a healthy breathing sound that the doctors hope she will hear. But if you're sick, it might actually sound like this. So this is an interesting fluid mechanical phenomena that happens in your lung when you're sick because you're breathing. And I'll also point out this is a project in collaboration with people from Michigan and a really fantastic graduate student that started this. So how do these crackle sounds occur in our lungs? It happens in the narrower parts of our lung airways, particularly when it's accompanied by surfactant dysfunction, meaning lack of biological soaps or surfactants in our lung. What happens is the liquid lining the walls of the airways will become unstable, form a liquid plug. And then when we breathe, the plugs will move and eventually rupture, leading to the crackle sound. So the doctors know this is an important symptom of disease, so we'll listen for it. Our question was, what effect might the fluid mechanical stress associated with this process, what kind of effect might that have on the cells? Could it contribute to disease or injury? Now this is a simple but important question, and it's actually a little difficult to ask or study in a systematic manner in the actual lung, because the lung is kind of hard to image, and the process of liquid plug formation and propagation, difficult to control and image in a real lung. On the other hand, traditional lung cell cultures in a dish also would not be able to create these interesting fluid mechanical phenomena. So this is where we thought, maybe a microfluidic device might be useful. So first, we developed the base microfluidic structure so that we could culture lung epithelial cells in a manner that would make them feel like they were in the lung. And we're trying to make here a healthy airway system. To do this, we made a microfluidic channel system with an upper channel, lower channel, sandwiching a semi-porous membrane. When it's put together, we can flow liquid from the bottom and liquid or air from the top with the lung epithelial cells at the interface. And this is a microscope image of the region where cells are growing. And this is a green stain that shows that the cells are healthy and happy. This is a close-up view, upper channel, lower channel, porous membrane. The cells grow here. Now, why is this device important for making the lung cells feel like they're really in the lung? Because in the lung, they have air on their apical side, the upper side, with nutrients supplied from the bottom, as they are here. And this culture we do for 3 weeks to make them behave and look like real lung cells, meaning that they have cilia that beat and secrete fluids and proteins like real lung cells would. Okay, so now we get a healthy lung mimic. Now for the pathologic pulsing flow, the liquid plugs that form. We need a mechanism to recreate this aspect. For this, our devices have, in addition to the cell culture region, a liquid plug generator device that looks like this. So the way this system works is we have an inlet with liquid and air flowing side by side. Periodically, we will shut the airflow off, filling the entire region with liquid. Looks like this. Now when we restart the airflow, air liquid flow will return to original with one additional thing happening, which is a little liquid plug gets formed and then pushed towards the cell culture region. Now for this liquid valving, switching, we use a solenoid valve. So this is a device description. So we have a solenoid valve that pinches a tube that is supplying the air. And that's how we switch the airflow on and off for this device. So here, we see this device. The liquid plug generator is actually on the right side, and you'll see a liquid plug come in. And that sound is a sound from a microfluidic airway. And the acoustic features look like this and are very similar to what is heard from a stethoscope from patient lungs. So now we have the fluid mechanical events that mimic pathology in the lung. So let's see what happens to the lung cells. So initially, the lung cells are healthy and green. But as we expose the cells to liquid plug propagation, and we do it at and expose the cells to different number of plug propagation and rupture events, what we observed was that we would see more and more red, injured, or dead cells because of the fluid mechanical stress involved. You could also see that the downstream regions are more red than the upstream regions. Because we have very well-defined conditions, we could study the fluid mechanical events going on in detail. This is by my collaborator, Jim Grotberg. Here's the airway, the liquid plug. As it moves, the liquid plug gets thinner because of trailing film of liquid. The velocity increases, and the pressures and stresses go up, explaining why there might be more injury in the downstream regions. So how bad can a little liquid plug be that you can hear with the stethoscope? If it's combined with surfactant dysfunction, which increases the surface tension, it can be a major cause of small airway injuries. So far, we've created a healthy airway, made a device that can recreate lung injury. So the final step is to see if we can treat it. So we demonstrated this with a similar but different device that, in addition to fluid mechanical stress, also generates the mechanical stress from stretching the lung cells. And here we see how the viability of the lung cells go down as we combine both stretch, which does a little bit of damage, plus liquid fluid mechanical stress, which then makes it go way down here. And this is something that mimics what happens in premature babies' lungs. And so also a picture of the damaged lung cells is kind of shown here. You can see kind of tears and red and so forth. So what do the doctors do in the clinic to mitigate lung injury in premature babies that don't have surfactants? Well, they will squirt into the baby's lung a therapeutic surfactant. And so we did the same thing with our lung on a chip, and we could show that we could rescue from injury when we did that. So we could, and this was done by other excellent postdocs and graduate students. So we've shown that using, by recreating an aspect of the lung, including pathologic fluid mechanical stresses, we could recreate a disease and also test some therapeutics as well. So that's my first specific example. Now, that was an example of where pulsing flow is kind of bad for the cells in our bodies. The next one is about pulsing flow that is beneficial. And this topic has to do with infertility, difficulty of having children. This is something that affects a lot of couples, millions in the US alone. There are many causes, including working too hard or too much stress, and the need for therapy is increasing. There are treatments, but it's expensive. There's room for improvement. Now, the cause of infertility is about 40% female factor, 40% male factor, 20% unknown. But when people undergo in vitro fertilization treatment, there is also factors that contribute to the infertility from suboptimal cell culture in dishes and other cellular manipulations. And so with my collaborator Gary Smith, we asked the question, can microfluidics help? So let me tell you about one specific problem we addressed, which is the problem of suboptimal culture of fertilized eggs before implantation back into the mother. So what happens in natural fertilization? The fertilized egg will spend its first 5 days being gently nurtured in the fallopian tube before it attaches in the uterus. Now, in vitro fertilization, the fertilization step, as well as the first important 5 days of someone's future child, is spent in a dish. And so imagine being an embryo. How would you feel being in a dish versus a nice fallopian tube? So we can actually quantify this by comparing day 5 embryos from a fallopian tube versus a dish. And these are mouse embryo data. And we can see the difference. So the mother-nurtured embryos have many more cells compared to the dish culture cells. And importantly, this leads to lower pregnancy rates. So could microfluidics provide some help in closing this gap? So we thought about fluidics, being microfluidic people. So what is the difference between the fallopian tube and a dish? There are many, but one simple thing we could think about is that there is a flow in the fallopian tube, both from cilia that beat and from contraction of the tube itself. So now we thought, we can address this question microfluidically. Let's see if we can develop a practical microfluidic system that can recreate some of these flows. And so some graduate students, postdocs, and actually an undergraduate student developed this system. So here's a microfluidic cartridge that has microchannels in it. And the material is made out of silicone rubber that's flexible, optically transparent. The embryo would be this big. And so the channels are actually smaller than the embryos. And what we do is attach these elastomeric microfluidic cartridges to something called a braille display. These are tools used by the blind to read email, but we use the piezoelectric pin actuators from these devices to pump and valve fluid in these channels. The system is all, okay, so here's how it works. Each pin, it can push and function as a valve, and if we have 3 or more pins moving in sequence, it's a peristaltic pump. And the advantage of having this type of system is we need the clinician to be able to introduce the embryo and retrieve it in a manner that's not too different from using a dish, and we needed a user-friendly format, and this was able to do it. Okay, so how do the embryos feel? Or how do they do? So this is a way to show the gap between conventional dish cultures versus what happens in the body for mouse embryos. So the y-axis is number of cells per embryo at day 5. And you can see a big difference. Now using the same culture media as in these cultures, but with dynamic microfluidic cultures, we could significantly improve the embryo development, although not quite as good as the real mother yet. We tested pregnancy rates and birth with mice, and here are microfluidic mice pictures. And we saw that the microfluidic cultures not only increased the number of cells per embryo, but enhanced pregnancy rates by 22%. And since we also have translated this to the clinic, and there are some human microfluidic births as well, and there are some evidence that the system actually enhances the quality of human embryos, not just mice as well. Okay, so what does this mean in terms of microfluidic circuit development? So in my first lung on a chip example, we used solenoid valves to switch flow one by one in tubes to switch the airflow, and that worked great. But for the embryo culture system, we needed peristaltic pumping, and we needed multiple pumps per circuit because we needed to culture multiple embryos. Now we could theoretically connect a bunch of solenoid valves, but that's kind of tedious and not convenient. And so what we did was to do parallel instruction and use the Braille displays to do microfluidic switching. However, this also still has limitations. We need an external brain, and in addition to the microfluidic channels, we needed external hands or actuators, the pins, to do the fluid switching and valving. And this seems clumsy compared to the electronic circuits that we have in our computers and cell phones and even simple things like calculators. So we tried to come up with something else. Or I should say, my student decided he wanted to do something else. So how many students are there here? A reasonable number. So I don't know if you've ever had this experience. You have a wonderful idea. You go to your advisor, and he or she says, I don't know if that's going to work. I'm not sure that's a good idea. OK, no one's laughing, so I don't think it happens here. But I do that sometimes. So this was an example. So Bobak came to me and said, you know, Shu, there are electronic oscillators that just use 2 transistors and some capacitors, and you can get self-switching oscillators. I want to try to do that with fluidics. And this is what he eventually came up with. So I told him, you know, that sounds like a fantastic idea, but no one's done it yet. I think if something like that would work, it probably would have been done. I haven't seen very much of it, so I'm not sure. So Bobak went off, not discouraged. And my students tell me he made like 50 design iterations after discouragement from me and came up with something that worked. So before the actual device, a little bit about electro-hydraulic analogy. So electronics and fluidics have a lot of analogy. So like in electronics, there's resistors. In microfluidics, there are channel and flow resistance. Electronics, there's capacitors. And in fluidics, you can also get something like capacitance when you have a flexible membrane, and when there's flow and pressure, you get deflection, and the deflected membrane can come back, and so that's kind of like a capacitor. So resistors and capacitors are very common in microfluidic circuits. How about a transistor? In fluidics, that would be like a valve or a switch. Well, things that function kind of like a transistor were actually common. A diaphragm valve that, depending on the pressure difference between this channel and that channel, would let flow through above a threshold pressure. But they were underutilized in microfluidic circuits, especially the small circuits. Larger hydraulic circuits actually used a little bit of this. So this is what Bobak came up with. So it's a little bit different than a transistor because it has like 4 terminals. But this is how this system that Bobak first developed works. So we have 2 microfluidic valves that are connected to each other. In the first, and I should say, so there's a syringe pumps that are pushing liquid from both sides. So in state 1, one of the valves is open, and so the green fluid flows through the top of this valve, through the bottom of the other valve, and then flows out. Because the bottom is pressurized, the red fluid doesn't flow, and it's stopped. After a while, though, the pressure builds up, and the red breaks through. Now the red fluid flows to the bottom of this valve, stopping the green, and the red flows out. And then after a while, the green reaches a threshold and goes through. And then it repeats itself. And so here's an actual device, and you can see it oscillate. If we attach pressure sensors, the pressures will go up and down in an alternating manner as well. And here's Bobak. He's now a professor at Cornell, and he made a variety of other circuits, including like this one, which is a timed switching device for performing immunoassays with sequential incubation and washing steps. So we thought this was great. Let's use this for biological experiments. Now, what's required for biological experiments are replicates, meaning we need to do experiment not just once, but multiple times. So how about doing replicates of microfluidic pulsing flow systems? Well, it turns out if you try to do that, as hard as we try to be perfect in our fabrication and operation, there's always variability between the oscillators in terms of its frequency, and aligning the phase is difficult as well. And each one has their own syringe pumps pushing. So we had to think of a better way to synchronize the fluidic oscillators. And so another student, Sasha Kai, came up with the idea, oh, let's couple the fluidic oscillators with fluidic capacitors. And so he did that, and he showed that he could get much nicer synchronization. The frequencies and the phase align better. Also, interestingly, we observed that the noise is less compared to when the oscillators are on their own. This is actually a known feature of coupled oscillators, but we could observe that also in these fluidic coupled oscillator systems. So we thought that was interesting because it's kind of a emergent property, or things that you only see when multiple components are coupled together. And so this is Sasha Kai, and he just got the cover of the journal, and I like this figure because it shows an actual device. This is a 2-oscillator system coupled with capacitors that look like this, and here's the circuits, and then a coupling capacitor, and it's synchronizing. So what does this mean? So for the embryo culture system, we developed a parallel instruction system that had that still had external actuators and an external computer to tell it what to do. And so as we want more valves, we need more external actuators, and we still needed to get external instruction. And it needed electrical power and actuation. What we showed with these fluidic logic circuits is we're flowing cell culture media or immunoassay reagent fluids into the device anyways. And because of the way the circuits are arranged, the fluid flow itself drives a logic circuit that self-regulates its flow. And so we thought this was perhaps a more scalable, useful method with embedded instruction in the device that's more similar to electronics. So how scalable and versatile do you think this is? So we thought initially, okay, we got it made. It's wonderful. However, we had problems. This was still not quite scalable and not free from electrical power as we would like. First of all, it needed syringe pumps to create the flow. But we could think of other ways to flow it. That was a minor problem, actually. More fundamental was the need for pressure fluctuations for this circuit to work. So this is in electronic, it's like having a constant current circuit, not a constant voltage. And for the oscillator to work, we needed these huge voltage surges or pressure surges for it to function. And the limitation of this is if we wanted to use one pressure source to operate multiple circuits, multiple oscillators in parallel. They all had to be going at the same time. We didn't have very much flexibility on what we could do with parallel circuits. So we wanted to get rid of these. And it took a few years, but a postdoc finally came up with a system that works like this. So this circuit has a slightly different design, but now it works with just gravity-driven flow. So there's just some height difference between the reservoir and the chip, and then chip to the waste outlet. Using this gravity head, this circuit has several of these transistor switches, fluidic switches, and now we can have multiple different frequency oscillators operating in parallel from the same flow source. And this is actually what we call a heartbeat chip, because it has 4 different frequencies, no pulsing, physiological resting pulsing, really fast pulse, and really slow pulse. And we have it at 3 different shear stress levels that mimic the capillaries, arteries, or something in between. And this is the postdoc Sunjing. And when he cultured blood vessel cells in these channels, he could see that the cellular response, there was a cellular response that was affected by shear stress level, which is the amount of flow, which was known, that more flow is usually more responsive of the cell. But another interesting feature that he saw was that regardless of the shear stress level, there seemed to be a frequency at which the cells responded the best. And so that was biologically interesting as well. In terms of device, we were very excited because this is now something that we feel is scalable in terms of operation. Whoops. I see it on my computer. The projector went off. Did I touch something?
Oh, there.
Oh, okay. Because it's a constant low pressure driven circuit. And so, we dream. So in the electronic world, went from very simple transistor integrated circuits to our phones and computers and so forth. Right now, we're still 6 to 8 component fluidic oscillators and switching systems. We don't aspire quite to billions of transistors, but we do aspire to medium scale integration and development of parallelized analog circuits. And using just conventional photolithography and microfabrication, we have developed some devices with hundreds of components. And now we're trying to look into 3D printing and other methods where we feel like some of these analog microfluidic circuits might be interesting as well as useful. Okay, so it's 15 till, what time do I have, Joe? 10 minutes? Okay, so I have one more topic on nanofabrication. And it's also a story about how experiments that go wrong can sometimes be useful. So I don't know if experiments go wrong in your lab, but we have a lot of failed experiments. So this was one of them. When we make microfluidic devices, we do plasma oxidation of the surface to activate and then allow bonding of pieces together. However, if you oxidize too long or mishandle your devices, the surfaces can become brittle, oxidized surfaces can become brittle and generate cracks. So this is usually something that's unwanted. However, one day we thought, how about if we intentionally do this with careful stretching? And we found that we could do pretty nice parallel cracking. And this is my first graduate student at Michigan, and she worked on this. Subsequently, my collaborator Michael and her graduate student developed a variant of this method where we oxidize a microchannel device and a flat membrane or microchannel featured PDMS mold and a flat thin piece, oxidize them, bond them together, and then stretch, in which case we get nanochannels. So here is a picture of a close-up of one region of such a device. So here's microchannels, microchannels, and in between, there's nothing right now. But if we stretch, we can create parallel cracks, tunneling cracks, which are actually nanochannels. Now, the interesting feature about these channels are that they are normally closed. If we don't, even after we stretch, if we let go, the cracks heal, and so there's no nanochannel, and the resistance is very high. Only when after we re-stretch again will the channels open, allowing current to flow and the resistance to go down. And then if we let go, it'll go back again. So using just convenient macro scale stretching and unstretching, we could have nanochannels that are not only easy to fabricate, but they have tunable cross sections that can be completely collapsed. So what do we use this for? We use this to map or image DNA and chromatin. So DNA, or DNA with its associated protein chromatin, is usually coiled in solution, so that even if you have a label, it's difficult to analyze what's going on. It's much more convenient if you could stretch out this DNA, and now you can see where the labels are. And if you have multiple labels, you can map your DNA. There are several ways to stretch DNA. One of the known methods is to use a nanochannel. Now, it's been known that if you really want to stretch the DNA or chromatin out extensively, you want to use as narrow a channel as possible. However, as a channel becomes narrower, it becomes harder to introduce your biopolymer into the nanochannel. If your nanochannel is big, it's easy to introduce, but then it's not as well stretched. So we thought, maybe our cross-sectional size adjustable channels might give us the best of both worlds. And so here's what we did. So here's the channel in its widened state. We can suck in the chromatin or the naked DNA and then squeeze and stretch them out. So easy loading, but then extensive linearization. Here's how it works. We suck the biopolymers in by widening the channels, and then as we narrow, the DNA in this case stretches out by both hydrodynamic forces and nano confinement, and then we can even go down eventually so that it's the channel is collapsed and the biopolymer immobilized. And here is an example of, so this was DNA, almost completely stretched with no electric field, just a cracked channel. And here we have chromatin, which has DNA as well as histone proteins. And we do multicolor labeling of different histones or different modified histones. And we could do 3-color mapping at least of these linearized chromatin fibers. And this is the postdoc that led this work, and all because of some failed bonding experiments. And I want to, and what we're trying to do next on this project is, so this is conventional fluorescent optical microscopy. Now we have linearized DNA and chromatin that is immobilized, which makes it amenable for super resolution imaging. And so the EBB microscope core just recently got a Vutara D-Storm super resolution microscope. And Aaron, the facility manager, is an expert on this machine. And there's also a fast confocal 2-photon microscope nearby as well that we're trying to use. And I think it's interesting potentially for many of you to use as well. And some of the students and postdocs currently working on this. So with that, oh, I have one more slide. I have an advertisement slide. So if you do things that are a combination of micro nanotechnology, biology, please consider integrative biology for submission. And with that, thank you for your attention, and happy to take any questions. First which experiment? Oh, that one? That particular movie was PBS. Saline, aqueous saline, with and without surfactant.
How do you differentiate between the damage that might be caused by other pathogens versus just shear mechanical stresses?
Well, so this device, so that's a really good question. So here I just showed you what happens in response to fluid mechanical stress alone. So I think another way to think about it is, okay, what happens when you also have bacterial insult or other inflammation and or immune response? And that's exactly what we're working on now. So if you have ideas, let me know.
Shu, thanks for the talk. So first of all, I'm a firm believer that if you get stuck with a research problem, you hire a new student who doesn't know what supposedly doesn't work, and he can solve the problem.
Fantastic. So, thank you.
Fully agree with you. So the question that I have is regarding kind of process optimization. So when I look at your different examples, for example, the fertility example, you showed that one bar there which had that improvement in, I don't know, what in
cell numbers,
cell
yeah,
size.
That one.
So do you do also kind of process optimization, flow optimization, frequency optimization in order to optimize that? And
yeah, so that particular one, we also did do some flow optimization, although we had kind of a target flow because so making the device is the easy part for those studies. It's like getting the animals and biological studies, that's the hard part. And so we can't do too many. And so we did have a target flow rate based on what people have measured as physiological frequencies and flows. Plus we had a preliminary study which seemed to show an effect with that flow rate. But we did do, so for the full statistical analysis with multiple replicates and 90 embryos, we did one flow rate. But to get there, we did preliminary studies with smaller groups with a few other flow rates. Yeah.
So I'm a sensors person. Right?
Uh-huh.
So my true question then becomes is, could you do this optimization with the help of sensors that monitor not the ultimate outcome, but something between that helps you optimize the system?
Oh, absolutely. So, so a lot of the things that we and others in the organs on a chip field have worked on is just how to make the devices grow cells. And now the entire field is getting to the stage where, okay, we can grow and do a lot of things, but how do the cells really feel? How are they functioning? And so sensing and analyzing, getting information out of the systems is becoming the key challenge.
The microfluidic oscillator that you talked about, I assume that it has some resonance frequency that you utilize. How controllable is that? What is the, how fast you can make it happen?
So, yeah, so I think the fastest we've done is about 10 hertz. It depends on both the components as well as the flow rate for the, or pressure. So flow rate for the first set of microfluidic systems and pressure for the second type. And we can make it as arbitrarily slow as possible. So for biology, although we have to have like resistors or check valves or something to do that. So for biology, I think in computer you want to go faster and faster. We talked to biologists and they wanted like, oh, that's too fast, can you do it slower? So we did slower. But the range we've done is between 10 hertz and like 0.00001 hertz, just because of the materials and dimensions we had.
Thank you. One more question. In electronics, we have amplification as well.
Ah.
I assume that here you probably inject more fluid, or how do you amplify it?
So there are people that have made kind of an amplifying system where they use small pressure to regulate flow of big pressure. In our system, we aren't doing amplification. And kind of related to that, compared to electronics, the resistance is really high in these, or at least it drops pressures and things a lot. And so we do have limitations in like how complicated or how small we can go because of resistance as well as size of cells or things that we want to do. And so we definitely have much more limitation in the complexity of the circuits we can make. And that's one of the reasons we lean towards more analog circuits than digital in terms of the circuit architectures and things like that.
Maybe one more question if there is any. Okay. Oh, go ahead.
What are the scales that you're talking about? What are the sizes of the cells, the fluidic pathways that you're making?
So the bacteria is like a micron or two. The mammalian cells are tens of micrometers. And then I guess the nanochannels are like going to like nanometer or something. So it depends on the biological material. But it's for living cells, it's microns and larger. For DNA and histone is like nanometers.
Walter, one last one.
Sure. So I'm just curious on the circuit equivalent question. I mean, in electronics, if you've got inductance capacitance, then you get a natural resonant frequency, and so you wouldn't necessarily have to engineer. Is there anybody looking at a fluid equivalent of inductance?
Yes, and so fluid flow momentum is kind of like inductance, so there is an I for fluid flow as well. In microfluidic circuits, it's typically negligible because the volume and amount of fluid with momentum is small. However, actually in our lung liquid plug chip, we had like fast flow and fast switching, and we did see small effects of inductance. So you can see it, but not enough to like generate oscillators based on that, at least if you want to stay small.
Let's thank Professor Takayama one more time.